talk about today is really some recent results from our lab that have defined a rigidity sensor that is involved in basically pinching the substrate and to a constant distance in measuring the force and thereby the rigidity. And the surprising aspect of this is that we find that that controls life and death on soft and rigid substrates, and that it is missing in virtually all cancer cells that are transformed. And that was a surprising result. So we've looked now at a variety of different cells, and all of them seem to be missing this complex, which is about 2 micrometers in size, contains many components that I'll talk about, but when it is missing, cells don't know the rigidity of the substrate and therefore grow. And that surprising result, we can toggle back and forth in a variety of different cell types by simply removing or adding back the respective cytoskeletal proteins that are missing. And in the absence of those cytoskeletal proteins, the rigidity sensor is missing and the cells are able to grow on soft surfaces. So going back, we've been interested in really understanding mesoscale aspects, i.e. subcellular mechanics. And the phenomenon that I'll be talking about today is really illustrated by this periodic contraction of the cell edge every 24 seconds, and the cell is sensing the rigidity of the substrate when it undergoes those contractions. That's analogous to this motor because it has thousands of components, and force, position, and timing are critical elements for it to function as for the automobile engine. And further, we find that it's present in a variety of different cell backgrounds. So like the automobile engine, it will work, which works in a variety of different cars. The rigidity sensing contractions will work wherever they're found. And it's really not sufficient to know the components present. It's much more important to try to understand how this system is actually working. This is a critical element in terms of differentiation, so that the studies from Disher's lab has shown that indeed taking mesenchymal stem cells and putting them on soft or rigid surfaces will drive the cells down a pathway of differentiation toward neurons if they're soft and bone if they're rigid. And we can see that when putting cells on top of pillars here that are tall, i.e. soft, or short, i.e. rigid, that the cells take on a different morphology at very early times. In other words, they're able at early times to measure the rigidity of that substrate. So that means that we can look at early times, and certainly fibroblasts are capable as well of measuring the rigidity of their substrates. And so we can use that system to really try to understand the process of rigidity sensing. And what was instrumental in all of this was really choosing the right phase of or state of the cells in this situation. If we take cells that are in suspension, they initially interact with the surface and then undergo a stereotypic behavior where they start to spread. During this phase of spreading, they don't measure any aspect of the surface. We can put them on passivated surfaces, and once they start spreading, they continue to spread over fibronectin or a passivated surface that they're not interacting with at equal rates. And then once they run out of membrane as they're flattening to a disc, a tension rises in the plasma membrane, and they transition to this contractile phase where they're measuring the substrate rigidity, and ultimately they will polarize, and whenever they extend a foot, they will measure the rigidity when they do so, but on average, this happens only about every 5 to 10 minutes. So the issue that was very important in terms of our initial aspects of understanding this process came about through a collaboration with engineers, and they were doing nanofabrication studies. And so we asked them to come up with a variety of surfaces with different diameters of pillars to see if there was any effect of pillar diameter on the behavior of the cells. And with 2-micron pillars, the adhesions formed around the cells, with one somewhat, but with 1/2 micron pillars, the adhesions formed on top of the cells, and myosin was between the pillars, pulling them together and forming what appeared to be local contraction units on the surface. And this was very similar in morphology to what we had on a regular 2D surface. So we followed up on this and found that indeed, as I was mentioning earlier, this process of spreading involved on stiff surfaces these periodic contractions. On soft, the cells didn't spread further, so indeed we believed it was part of rigidity sensing. And when we used the 1/2 micron pillars, we found that sure enough, near the edge of the cell, we could see these contractions as an inward pull, which is in red here, and an outward pull, which is in green, and these were separated by about 2 micrometers, as I suggested earlier. And the other surprising aspect of this is that when we look at the length of the arrows here, which are indicative of the degree of displacement, the red arrows are larger than the green arrows, so there are greater forces applied inward than outward, which fit with the idea that there was flow of actin inward in this region of the cell, which has classically been known, and indeed this rigidity sensing scales with the rigidity. So in other words, they pull to the same length no matter what the rigidity of the substrate. They increase the force on very rigid substrates. So when we put the cells down on more rigid pillars, it was more like 47, 43, indicating that indeed the flow was the important issue. So if we look at those individual contractions here, we see that they're transient, which is a little bit surprising, but they pull to, in this case, a length of about 60 nanometers in an anti-parallel fashion for about 30 seconds, indicating that it was like you or me making a transient pinch of the surface and thereby measuring the rigidity of that surface. When we analyze this in more detail, a guy, Wolfenson, got a 100 hertz camera so we can measure the position of the pillars to within about 1 nanometer every 10 milliseconds. We averaged those frames here for 15 frames, and then applied a step analysis program and saw that there were real steps in this contraction, indicating that myosin was pulling this and stepping along the actin filaments. This is the control, taking that slope and adding noise to it, and we analyzed that, and that gave steps of about 3 angstroms, but the major real steps were about 1.2 nanometers. This still begged the question of whether we were seeing contractions toward one another as would be predicted for a true sarcomere-like unit. And when we analyzed some in detail, we could see here in the orange this step was moving inward. At the same step, the green pillar was moving outward, and the difference on average in terms of the time between those steps was a Gaussian around 0. Two other pillars in the same region that were pulled in the same directions were not coordinated, and the overall displacement here is about 2.4 nanometers, which corresponds to the distance of half of an actin monomer along a single protofilament, or the distance going from one actin monomer on one side to the next actin monomer on the other side of that filament. So this seemed to be a geometric issue and begged the question of what was really involved in rigidity sensing. And so Hagai carefully analyzed the data and found that indeed after about 4 to 5 steps on the rigid pillars there was a pause of about 2 seconds, and on the soft it waited for about 10 to 12, so that these were about the same force, about 25 piconewtons, and this was activating, it seems, some pause in this contraction. And when we looked at what happened during that pause, there was indeed a biochemical correlate in that this is the pause, and we see that alpha-actinin is accumulating at those pillars during that pause and starting to really accumulate. It accumulates a lot faster than the overall displacement here. So we see a peak in the amount of alpha-actinin around a pillar before the force on that peaks, a difference of about 7.5 seconds. So this now begged the question of how is indeed the force being sensed? 25 piconewtons is about the force needed to pull a lipid out of a bilayer, and certainly enough to unfold talin to a length of almost 1,000 nanometers. So we didn't see any change in length here. So this was rock steady at about a nanometer, constant distance. So the model that we have, which is at this point pure bullshit or speculation, let's put it that way, is that the membrane proteins are bending and bringing enzymes near the surface and thereby catalyzing, for example, PIP3 production, which is then recruiting alpha-actinin. So there's more studies that I won't have a chance to talk about today in detail. Came out in a recent paper, Saxon et al. Nanoletters, showing that another element of this sensing process is the cleavage of talin and the release of the talin rod, which aids in this contractile unit formation. We don't know quite how, but does indeed and is necessary for growth in the system, even in the presence of intact non-cleavable talin. So I'm going to switch gears and talk about one of the really interesting aspects of this process, which was the slow rate of contraction of these pillars, which enabled all these measurements to be made. And it's about 2 to 3 nanometers per second that they're moving toward each other. If you take myosin 2A, which is the myosin driving this, in vitro, it moves actin filaments at 200 plus nanometers per second. So it's working at about 1% of its active rate in this situation. So why is that? Perhaps it could be slowed down by tropomyosin, was our initial thought. And so Hagai went, looked and stained in that region of the cell for tropomyosin and found that there was one isoform of tropomyosin that is then known as tropomyosin 1, now it's known as tropomyosin 2.1, and he knocked it down. And sure enough, he found that when it was knocked down, the velocity of contraction increased, step length increased, the overall displacement of the pillars on soft and rigid increased, the surprising finding was that the cells no longer sensed rigidity. And they moved the same distance or spread to the same area on soft and rigid, whereas the controls spread much more on rigid than on soft surfaces. So we followed up on this, and it turns out that Peter Gunning had beaten us to this and knew that the tropomyosins were important for at least cancer cells in terms of their rigidity sensing. And they had correlated this tropomyosin in particular with cancer and showed that in virtually all the major cancers looked at, that this tropomyosin was missing or downregulated. And sure enough, we take MDA, MB231s, the human breast cancer line, and they miss tropomyosin 1 at the edge. The controls, MCF10As, have it there. Studies have been done to show that adding back tropomyosin to MDA, MB231s caused them to be non-transformed, non-metastatic, I should say, and behave normally. So when we did the converse, we took tropomyosin 1 and depleted it from MCF10As. They now grew on soft surfaces, indicating that it was sufficient to remove or add this single cytoskeletal protein to drive a cell from a transformed to a non-transformed state. So that was a sort of a crazy idea that simply modifying a cytoskeletal protein could cause transformation. So we tested it out again with alpha-actinin, and alpha-actinin 4 is part of this contractile complex. Knocking it down removes the contractile complexes, and the cells now are able to grow on soft surfaces. Controls are not. So that raised the question about actual cancer cells. So we tested 4 different cancer cells that are all transformed, as well as COS7 cells, which are not cancerous but are indeed transformed, and looked at the number of local rigidity sensing contractions that those cells would exhibit in the first 10 to 20 minutes on a surface, and found that none of those cells were able to generate a significant number of sensing contractions, whereas the HFS gave 10 to 100 times as many. The various ways in which those cells were transformed are quite different. The COS7s are SV40 transformed. The MDA, MB231s are KRAS mutated, NRAS for the HT1080s, and P53 mutated for the ovarian cancer cells, and the mouse-Lewis lung are really sort of messed up. So the question then was what proteins might be altered in these cells, and we looked at the proteins associated with the rigidity sensor. This included some of the receptor tyrosine kinases, which we found were involved in the rigidity sensing. Those were not, or were seemingly altered in a haphazard way. COS7 were missing myosin 2A. MDA, MB231s were missing tropomyosin 2.1, this lower band, and as was SKOV3s. And the HT1080s had 2.1, but had a very high concentration of tropomyosin 3 that Gunning's lab had correlated with its overexpression with a variety of cancers. So the next issue was whether or not we could bring these cells back into a normal state by restoring these cytoskeletal components as opposed to manipulating the tyrosine kinases. So with the COS7s, they were missing myosin 2A. We added back myosin 2A, and now they seem to pull equally well on soft and rigid surfaces. This is the maximum displacement of the pillars. And they now had rigidity sensing contractions. And if we now took and depleted tropomyosin 2.1 in those cells after they have been restored to a normal behavior, we found that they pulled much more on both rigid and soft surfaces, and they no longer had rigidity sensing contractions. In terms of their growth and anoecus, or cell death on soft surfaces, when we restored myosin 2A, they activated caspase on soft surfaces. They didn't grow on soft agar, whereas the wild type missing myosin 2A did just fine. And when we put back the myosin 2A, if we further deleted tropomyosin 2.1, they now grew well on those soft surfaces. Doing the converse type of experiment with the MDA MB231s, we found essentially the same type of behavior, namely that they started out pulling very hard on the surface, and if we now added tropomyosin 2.1, they pulled less and equally well on soft and rigid. They had the rigidity sensing contractions, and we could abrogate those or get rid of those by depleting myosin 2A here. They didn't pull very well and didn't form those rigidity sensing contractions. But in terms of growth, they now would grow in the absence of myosin 2A on soft surfaces, and they were no longer activated caspase in the anoecus pathway, which was excited by the re-expression of tropomyosin 2.1. So we've tried a number of others as well, the SKOV3s, which were missing tropomyosin 2.1, again, are restored to normal behavior by adding back that single-sided skeletal component. The question that this all begs is whether or not the tropomyosin 3 is involved in this process, because the HT1080s have 2.1, but they overexpress 3. Conversely, MDA MB231s have no tropomyosin 2.1, and they have a low tropomyosin 3, but still significant amounts. So what Beverly Yang did in this study was to knock down tropomyosin 3 in the HT1080s and was able to restore the rigidity sensing contractions on both soft and rigid surfaces, but not with the MDA MB231s because they lack tropomyosin 2.1. So coming back to my initial slide, what this tells us is that it's not the receptor tyrosine kinases that are altered, it seems, in transformation, but indeed the ability of the cells to have rigidity sensors, and if you take out that rigidity sensing unit, then they are transformed. And we know that the RTKs are involved. They seem to be involved mechanically here. The axle sets the length of the contraction. ROR2, another receptor tyrosine kinase, sets the length of contraction. It binds filament A. Axle binds to myosin and in the middle of the myosin filament and to tropomyosin. And this is all activated by EGFR and HER2 in a SRC-dependent manner. So all of these receptor tyrosine kinases in this function appear to be involved, acting without ligand and are acting as mechanoregulatory components. As I say in my title, the rigidity sensor will activate growth on a rigid surface through the production of the talon rod and by calpain cleavage. On the soft surface, and this is now work that's just being written up, it seems the talon head is involved with PTPN12 or PTP-PEST in activating death-associated protein kinase to cause death. And I've drawn it here. DAP kinase is associated with this complex and indeed phosphorylates tropomyosin 2.1, and that phosphorylation is critical for the assembly of this complex. So we have a lot more to learn about this, and I welcome you to come and help us out with this. This is also, it seems, something similar is happening in terms of cell-cell interactions because cadherin-coated pillars here undergo similar local contractions toward one another, collaboration with Benoit Ledoux. So my major take-home message here is that we're used to thinking about self-driving cars, and the important aspect of those is that they have sensors that tell them about their local environment and then cause them to stop, presumably when something gets in their way. This is what happens when you're going at 70 miles an hour and you're watching a movie and a semi-trailer comes in front of you and you don't have the proper sensors to tell you about that. So indeed, the cells need those sensors to grow properly. Thank you very much.